Hello all, I'm Youngmin Son, who is an assistant professor in the Department of Systems Biotechnology. Before starting, I'm really appreciative to the boards of New Agro-Food Research Institute for giving me this great opportunity of research presentation in front of you. As you see in this screen, the title of today's presentation is Tissue-Resident Helper T cells, key manager of the local adaptive immunity against respiratory viral infection. Simply, this is about one novel CD4 T cell population in the lung. Recently, I and my former lab member found this interesting cell population, and then we figured out for their characters and functions. The contents are consisting like this. First, I'm going to show you the data about these novel cell types which contribute to the local adaptive immune response against respiratory viral infections like influenza virus. Second, this is still unpublished data and it is for the mechanism of this cell type maintenance during the long-term period. The title of first section is the role of tissue immune cell circuit in the immunological protection against respiratory viral infection. As I mentioned, I had used most animal models with influenza virus infection. So why we need to study influenza? It is very well known that the influenza virus is one of the representative segmented RNA virus. Also, that is can cause severe acute respiratory disease. From the October 1st, 2019 to February 15th, 2020, US CDC reported the case of patients who have some phenotype by influenza viral infection. As you see here, 29 million to 41 million people have flu symptoms. Then 13 million to 90 million patients met the doctor because of their severe disease. About 280,000 to the 500,000 patients were eventually hospitalization by their severe disease and their painful symptoms. Unfortunately, 16,000 to 41,000 patients were finally died. The reason why this number are critical issue in these days, because influenza is still one of the most critical virus cause severe respiratory disease, even though the vaccines are offline every year. So that's why we have to still keep going study the influenza virus. Now I would like to share some basic information about T-cell immunity. As you well know, T-cells are usually activated and expanded when they are recognized pathogen information by contacting with antigen-presenting cells like dendritic cell in the lymph node. The numbers of those activated and expanded effector T cells are sharply going down when they have done their job, like the killing the infection cell and the contribution to removing the pathogen. Finally, the real leftover cells are going to memory T cells. Those memory T cells are preparing the body protection against secondary pathogen infection. Now, let's talk about the tissue-resident memory T cell. What are the tissue-resident memory T cell? This is a quite new concept of memory T cell in the immunology field. Briefly, all we know that the traditional memory T cell like effector T cell and central memory T cells. As you've seen here, the effector memory T cell can circulating everywhere through the blood to non-lymphoid tissue. But Central memory T cell can only move around blood to secondary lymphoid organs like the spleen and lymph node, not going to non-lymphoid tissue. In this case of tissue-resident memory T cell, they can stay the only inflamed tissue which have experience of pathogen infection, and they cannot move out from these tissues. That's why we are calling these cells to tissue-resident memory T cell. The tissue-resident memory T cell, the TRM, were observed in a lot of different tissues, like the brain, lung, FRT, skin. So we believe that the resident memory T cell mediated the frontline immunity against reinfection in the peripheral tissues. Next, how we can detect TRM cells. There are several markers to identify the TRM cells in the memory stage. The most useful marker is CD69, which suppresses the cell egress out from tissue to blood, and CD103 is also one of the representative markers to distinguish the TRM cells. Usually, CD103 bind to each category of tissue epithelial cell. That's why they are holding and staining the tissue. The other CD69 alpha and CXCR6 are also used as a marker of TRM cell. So with those useful markers, we can detect and tracking the TRM cells in the tissue. Now we are wondering, how are those TRM cells important? First, when the same pathogen invaded into the cell tissue again, the TRM cell responds immediately and kills the infected cell. At the same time, they secreted the active cytokine to activate it and recruit the other immune cells like the B cell, CD4 T cell, and the dendritic cell. So TRM cell provides the superior and the protection to prevent viral spreading and severe disease development very rapidly. My former colleague, Wang Zhang, he reported the mechanism of CD8 TRM cells maintenance and the important protective function of TRM cells against influenza viral infection. He blocked the CD28 signaling, which helped the TCR stimulation from antigen-presenting cells to T cells. As you see here, this slide, without CD28 signaling blocking the B7 antibody, the certain epitope-specific TRM cells couldn't maintain at the memory stage. Also, the mice which have no CD28 signaling have an impaired body protection by secondary influenza infection. That means that the red graph, the anti-B7 antibody treated mice has a lower recovered rate. So this data suggests that influenza epitope, particularly MP-specific TRM cells, are required for the host protection against secondary viral infection. And also, other former landmate, Chung-Ang Lee, he found the key transcription factor BHLHE40, which involved with maintaining TRM functions following influenza infection. Originally, BHLHE40 was reported as a stress-responsive transcription factor involving the circadian rhythm. But when he impacted the influenza virus into the BHLHE40 knockout mice model, the antigen-specific TRM cell was significantly reduced, and the body protection was also significantly downregulated against secondary influenza viral infection. So we can learn that influenza epitope-specific TRMs are required for host protection against secondary pathogen infection. Together with those research articles, now we know that TRM cells are very important for the host protection. But as we have seen the previous data, most TRM papers are focused with the CD8 TRM cell only. How about other tissue immune cells like the CD4 T cell and B cell? 2019, one of the big guys in B cell field, Troy Randall, he demonstrated the importance of tissue-resident memory B cells in the local immunity. After that, a lot of related papers will comes out, even particularly the B cell. The one interesting thing is most of those tissue-resident memory B cells were observed in the lung after influenza virus infection. Now I mentioned about CD8 TRM and tissue-resident memory B cell, we call the BRM, after influenza viral infection. But still, there were very few reports related to tissue-resident memory CD4 T cell. As you know, traditionally, CD4 T cell have strongly have function to B cell and CD8 T cell. One special subtype of CD4 T cell, the follicular helper T cell, we call the TFH cell help B cell to form the germinal center structure. Then it also contributes the B cell differentiation into memory B cell. Other subtype of CD4 T cell, the TH1 cell type, assists the memory B cell, memory CD8 T cell functions. Those responses are one of the key players in the long-term immunity. But all of those responses were happened at the lymph nodes or spleen, like the secondary lymphoid organ. The roles of local CD4 T cell help in the formation of TRM and BRM at the mucosal tissue after resolution of primary viral infection are still unknown. To figure out the novel role of CD4 T cell, I have depleted CD4 T cells after effector stage. As you see in this imaging, the control group have a clear immune complex IBART like the atopic lymphoid structure in the lung, but I couldn't see any IBART structure at the CD4 T cell depletion group. At the similar experimental design, I have treated FTY720 reagent, which block the cell circulation to observe the only local event with minimizing the circulating cell effect. In this condition, the both antigen-specific CD8 TRM and BRM were significantly reduced, even though depleted CD4 T cell only. So the data suggests long CD4 T cells provide late help for the formation of BRM and CD8 TRM cell response in situ. Then, to identify which tissue CD4 T cell subtype have this unique function, the long CD4 T cells were sorted and then performed single-cell RNA sequencing. As you see here, interestingly, I found one follicular helper T cell-like population at the cluster 3. And to tracking this population, I have used the PD1 and FR4 as the specific marker. With these markers, I observed the clear cell population like this. For further analysis, PD1 high, FR4 high cell, and other cell population were sorted, then performed bulk RNA sequencing. When I compare long PD-1 high FR4 high cells to long PD-1 low and FR4 low population, the TFH-associated genes were significantly high enhanced at the long PD-1 high FR4 high cell population. The bottom graph, the TRM-associated genes were dramatically enhanced at long PD-1 high FR high cell than spleen TFH cell. So I had concluded a population of CD4 T helper cells in the lung mucous tissue exhibiting TFH and TRM features like the hybrid. The parabiopsy surgery is one of the golden standards to identify the feed tissue resident cells. So I have performed this parabiopsy surgery with influenza-infected congenital mice. The CD45.1 and CD45.1 and 2 double positive mice were influenza viral infected respectively. After 21 days post-infection, the mice were hooked up by parabiopsy surgery. Additional 14 days later, the PD-1 high FR4 high population were measured based on the CD45.1 single positive black host mice. Most PD-1 high FR4 high cells were marked CD45.1 single positive, but there were very few number of CD45.1 and 2 double positive partner PD-1 high FR4 high cells. I mean the red bar. It was shown a similar pattern at the partner mice as well. So I believe the long PD-1 high FR high CD4 T cells are real tissue resident. And then those tissue resident helper T cells were called TRH cells. As I mentioned at previous data, the long TRH cell exhibiting the features of both TFH and TRM cells. So I have impacted influenza virus into the BCL6, which is the key transcription factor of TFH cell, knockout mice, and check the TRH cell. As I expected, TRH cell were significantly reduced BCL6 knockout at the BCL6 knockout mice. And I also checked the TRH cell in the PHLH40, which is the key transcription factor of TRM cell, and this knockout mice as well. I can see the lower number of TRH cell in the PHLH40 knockout mice than the control group. So this data indicating BCL6 and BHLH40 both are required for development of TRH cell. Then, to make sure that the TRH cell is the real unique population to assist CD8 TRM and the PRM development and maintenance, I have designed the BCL6 conditional knockout mice with the BCL6 plus CD4 ERT2 system. In this system, the BCL6 was abrogated when I treated tamoxifen, so I can control the time point of BCL6 depletion. I didn't put the TRH cell number data here. The TRH cells were dramatically diminished in the BCL6 flux CD4 ERT2 mice model. And also, antigen-specific PRM and TRM were reduced together with the CD4 TRH cell. To figure out the functional effect, influenza virus were challenged at the same experimental condition. In those downregulated, the adaptive immunity group showed a lower body protection than control group. So this data means TRH depletion infers mucosal protective immunity against influenza reinfection. Next, I wonder how TRH cell help CD8 TRM cell maintenance at the late time point. One of the special characters of TRH cell is that they express higher levels of IL-21 cytokines than other cell types. As you see this data, the most IL-21 high expressing cells were TRH cells. I had tested the function of IL-21. IL-21 signaling was blocked by anti-IL-21 receptor antibody treatment. So without IL-21 signaling, MP-specific CD8 TRM were significantly decreased. When I did the secondary influenza virus infection, the virus titer in the lung was still quite high at the IL-21 signal blocking group. So this data suggested TRH cells have CD8 TRM by IL-21 dependent manner. Unlike to CD8 TRM cells, the PRM cells were not affected by IL-21 signaling. They were not changed at all under IL-21 signaling blocking condition. So next, to identify how TRH cells help PRM maintenance, I had focused on the CD4 ligand signaling because traditionally TFH cells express high levels of CD4 ligand, and also in my hands TRH cells express high levels of CD4 ligand on their surface. I didn't put the data here. So I treated the anti-CD4 ligand antibody. Then I found the germinal center B cell and antigen-specific BRM cells were significantly reduced. The result indicating CD4 ligand signaling is likely involved in TRH cell help-to tissue B cell. This is a summary. First, I found lung CD4 T cells are co-localized with the tissue B cell following influenza infection. Second, there is one unique CD4 T cell subset which is expressing PD1 and FR4. Third, those PD1 high, FR4 high cells are exhibiting both TFH and TRM features with high expressing BCL6 and BHLHE40. Last, TRH cells help CD8 TRM via IL-21 signaling and also help PRM cells by CD4 ligand interaction. These local immunological circuits contribute to the mucosal protective immunity. So my conclusion is that the TRH cell assists the development of respiratory protective CD8 and B cell immunity. Okay, now let's move on next section, the mechanism of TRH cell maintenance. These projects are almost done and now preparing the manuscript. One of my collaborators, Dr. Carolyn King, she published a paper about story of TRH cell at the same volume of journal with my one. At this paper, she described the BCL6 positive TRH cell were co-localized with the lung B cell, and without B cell or MHCRES2 signaling, the TRH cell was significantly decreased. But unlike two lymph nodes, lung atopic lymphoid structure like IBAT, there are different types of MHCRES2 positive cells like DC, macrophage, B cell. So still we are not sure whether the local antigen stimulation was caused by MHCRES2 signaling of B cells. To figure out this question, I have done several other experiments. First, I have reconfirmed the T cells of TRH cell and found it expressed a higher level of IL-21, BCL6, and CXCL5, which is similar to my previous paper. Based on my data, I have checked the location of IL-21 high-expressing TRH cell by comparative imaging. As you see here, as I expected, it is placed with the long pieces. The green light indicating the IL-20 expressing cell, and red means the CD4 T cell. And those IL-21 expressing CD4 T cell is inside of B cell structure. And the lymphotoxin beta is an important molecule to maintain the atopic lymphoid structure like lung IBAT. I treated anti-active lymphotoxin beta receptor antibody to abrogate the IBAT structure in the lung. As you see here, the anti-lymphotoxin beta receptor antibody treated mice group has no IBAT structure at all. And also, the antigen-specific TRH cell number was also significantly downregulated. This means IBAT structure in the lung is required for the maintenance of TRH cell. From the bulk RNA sequence data, I found TCR signaling associated genes were dramatically enhanced at the lung TRH cell than other cell types. Next, I checked the expression of NUR77, which is the TCR signaling downstream molecule. As you see here, the lung TRH cell express high levels of NUR77. This result indicating high levels of TCR signaling is processing in the lung TRH cell. To identify what kind of antigen stimulation is happening at the TRH cell, I have designed the artificial antigen stimulation into animal model. The CD45.1 single positive OVA specific OT2 cell were transferred into different congenning mice. Then, influenza virus, which including OT2 epitope gene, was infected. One group was got just a PBS, and the other group was administered with OVA every 4 days. Day 28 post-infection, I have checked the OT2 specific TRH cells. Very interestingly, continuous OVA administrating group have still very high number of OT2 specific TRH cells, but PBS treated group have no TRH cell at all at the lung. And this result were only observed in lung tissue. At the spleen TFH cell, there was no change because I treated the OVA through the intranasal route. I think this is one of the key factors. So this result demonstrated that persistent antigen stimulation are required for maintenance of TRH cells in the lung. The final question is which cell type is the main source for providing MH class II signaling to TRH cell in the lung tissue? To answer this question, I generated mixed bone marrow chimera mice based on the MHC2 flux UBC CRE-ERT system. The irradiated mouse group had received MHC2 flux UBC CRE-ERT bone marrow and mu-MT bone marrow by 1:4 ratio. Mu-MT mice have generally no B cells. The other irradiated mice group had received wild-type bone marrow and mu-MT bone marrow by 1:4 ratio as well. After 4 weeks, the influenza virus, after 8 weeks, influenza virus was impacted into each group. 12 to 14 days post-infection, I treated tamoxifen to abrogate the MH class 2 expression. After treating tamoxifen, the first group have only MHC2 depleting B cell. In this setting, I have measured antigen-specific TRH cell at day 42 post-infection. As you see here, the MP-specific TRH cell was significantly reduced at MH class 2 depleting B cell group. But they have a partial effect. So additionally, I also treated the effect of other contact-related molecules such as CD4 ligand and ICOS. The CD4 ligand and ICOS signal have an effect to control the TRH cell maintenance as well. So I would say TCL stimulation from MH class 2 of tissue B cell might be major source to maintain the TRH cell in the lung. This is summary for the second section. TRH cells are co-localization with the tissue B cell at the lung. TRH cells have been received the TCL signaling from the MH class 2 of B cell, tissue B cells. Due to this interaction, TRH cells can be maintained at the long-term immunity in the lung. All the data from first section was done by Dr. Son's lab, which is from the Mayo Clinic and University of Virginia. When I was postdoc, the Dr. Insu Chun and Dr. Li Chaopan, they helped me for finishing this first section data. And the second section part will be finished soon in my lab. So I'm really appreciative to your attention. Thank you.